talk about the three types of bonds: covalent, ionic, and hydrogen. You should know the difference between those three and be able to identify them. If I show you a picture, how do you describe a covalent bond?
Shared electrons.
Sharing of electrons. Is there any covalent bonds here? Guess where?
The O's and the H.
Where?
In the middle.
Good, yes. Right here, between the hydrogen and the oxygen within the water molecule, these are covalent bonds. Same thing over here with this oxygen and this hydrogen, and this oxygen and this hydrogen. These are covalent bonds. What other type of bond can you see here? So obviously it's labeled, right? The hydrogen bonds. Those are the dotted lines, the weak attractions. We said opposites attract, so we'll have a partial positive charge attract to a partial negative charge. And this what links the water molecules together. We talked about a bug floating on the lake, for example. So we were putting atoms or elements together to make up compounds. We could take hydrogen atoms, add oxygen atoms, and we would make water. We have one substance, we add another, we get something different out of that mixture. This is a chemical reaction. Basic chemical reaction, we could say, is A plus B produces C. I've heard that before. The A and the B, this is my A, this is my B, these are both called the reactants. They're the substances that are going to react with each other. Okay, the C is the product. And I said last class, chemistry is like cooking. You're going to mix butter and sugar and eggs and flour, and those are all your reactants. And then what are you going to get at the end? A cake. Right? That's your product. This chemical reaction is moving in one direction, and you can see that from the arrow. We can take A and B and make C. If the reaction is a reversible reaction, then the arrow that is shown here going towards the products will be reversed, and you will see it pointing as well towards the reactants. And that means that you could break this substance down, digest it or decompose it, break the bonds here in the product, and get the original reactants back. Sometimes we can do that, sometimes we can't. We can't really take the cake totally apart and get right back to the sugar and the eggs the way that they were before. Okay? But with many chemical reactions in the body, we can essentially do just that. So if you take a look at this reaction here, you can see what it looks like if we were able to take apart C and get back to our original reactants again. In this case, C now is our reactant, and the A and B are our products. Is everyone with me on that?
Mm-hmm.
And you just have to look at the positioning of the arrow. That's telling you which way the reaction is being driven. As we are breaking down C, this breakdown reaction is called a catabolic reaction. It's also called a dehydration reaction, decomposition reaction, where we're decomposing C into A and B. So this is a decomposing reaction, digestion reaction. Whereas the first one that we were looking at, where we're taking A and adding B to get C, that's a synthesis reaction. We are making something. It's an anabolic reaction. I think we talked about this maybe the first class, where I might have said ana make and cata break. Anabolic reactions make, synthesize something, and catabolic reactions break something down, decompose something. We have a combination of these reactions occurring all of the time in our body, anabolic and catabolic reactions, and the sum of all of these reactions is what? Good, metabolism. Remember that term, very good. Okay, metabolism. It's the sum of all of these reactions in the body. Often when we look at the synthesizing of a substance, the making of a substance, that reaction involves the dehydration of the reactants. And dehydration means what? What are we doing? We're taking out water. We're removing water from a substance. In order for A and B to come together to make C, we need to dehydrate those two reactants. We need to take water out of them. And water, as you know, is H2O. So we're going to take out two hydrogens and an oxygen from A and B. Okay. We might take a hydrogen and an oxygen from A and just a hydrogen from B. You with me? Yes? Or we could take just a hydrogen from A and a hydrogen and an oxygen from B. Either way, we're taking out two hydrogens and an oxygen and dehydrating those two substances. So at the other end of our equation, as a product, we're getting water. Aside from the product that we're producing, which is C, we're getting water out of it. Because we've taken those substances and taken the water out of them. Okay. We call this type of an anabolic reaction a dehydration synthesis reaction. In order to synthesize something new, in order to synthesize the C, our new substance, we need to dehydrate the A and the B. And think about it in terms of the atoms and the electrons and how they want to be stable. So if we're removing a hydrogen from one and an oxygen and a hydrogen from the other, we're changing the stability of those atoms. Yes? Mm-hmm. So now they're not really stable anymore, and they're going to want to do something to gain their stability. They're going to want to bond. And this time, rather than them being separate entities, they're going to bond with each other. And so it's a way for us to allow A and B to then come together to make C, to be able to bond. So we took those hydrogen and oxygen atom out in order for those substances to want to bond together. Is everybody with me on that? You'll see examples of this as we go through the class today. As we talk about the organic molecules and how we can put them together to make larger organic molecules, you will definitely see examples of this. Yes?
So you're removing the water from A and B,
yeah.
Then where does it go if it's not going to C?
It's not going to C, it's just collecting within the body. And essentially water is one of those products that we can then break down into hydrogen ions, and they're going to accumulate on one side of the membrane, and they're going to help with aerobic cellular respiration.
So you like essentially have
Save it.
You're having two products. You're just
Right. And the water we call a byproduct. So it's not really the main product we were looking for. We want the C, but it just happens to be another product that we make. So we often call it the byproduct. Questions on this? So if you look at the C being broken down into A and B, at this decomposition reaction, where we're breaking C down now, remember water was a byproduct from the first series of reactions? With this one, we now, in order to break C, which is really A and B stuck together, in order to break them apart, have to put water in to that reaction. We have to add water to C to force the A and B apart again. This is called a hydrolysis reaction. Look at the word, hydro and lysis. What is, well, I wrote it here, but lysis means to break something. You lyse a wound, or you pop a balloon.
Red blood cells.
Lyse something. So we are breaking down water. We're breaking the bonds within water. We're breaking those covalent bonds, linking the oxygen and the hydrogens. When we break those bonds by adding the water to C, then the hydrogen and the oxygen need to go somewhere because they're no longer being held together as water. Where are they going to go? They're going to force the C apart, and they're going to bond back to where they were before, in the A and in the B. With me? Yes? So hydrolysis is a decomposition reaction where we're adding water essentially to break the water apart to digest the substance. Think about this as when you eat, you get thirsty. Right? It's difficult to eat for a period of time without wanting to drink something. Your body's telling you, well, if you want me to digest all these C's, you better give me some water in order to do that. So this is digestion. That's why you get thirsty. Redox reactions are what we call coupled reactions. So if we were looking at A plus B goes to C, that's our one reaction. But at the same time, we were looking at F plus G equals Q. And these two reactions occur exactly at the same time and really depends on each other. Then that's what we call a redox reaction. The re stands for reduction, and the ox for oxidation. So one reaction is a reduction reaction, where we're reducing the substance, and the other reaction is an oxidation reaction. In an oxidation reaction, we lose electrons, and with it goes hydrogen. So if we lose hydrogen, we're assuming we're losing electrons, that's an oxidation reaction. We're oxidizing the substance. The substance in this particular case will typically have a charge on it. And since we've lost that hydrogen, you might see a negative charge there. Okay? You might see a positive charge there. But you will see a charge. In the reduction side, when we're reducing a substance, we are gaining a hydrogen and we're gaining an electron. So you'll see an addition of an H at the end of the chemical formula, or an addition of an H2 at the end of the chemical formula. That will show you that you're looking either at reduction or you're looking at oxidation. We have two reactions occurring at the same time. One of them is going to lose some hydrogen and electrons, and in the other reaction, we're going to gain them. It's kind of like the ionic bond that we talked about last class. Remember the ionic bond? We had a donator and we had an acceptor. It's basically the same thing happening here. We have two reactions. In one, hydrogens and electrons are going to get lost. Where are they going to go? They're going to go over here, right, to this reaction. We will talk more about this when we get to cellular respiration in a later chapter. Actual chemical reactions of glycolysis and the Krebs cycle and electron transport, about 105, you should know what I'm talking about. We won't get into a lot of the details until A&P 2. At least you know, you're familiar with what a redox reaction is. Losing electrons and hydrogen from one reaction and then putting them in to another reaction. Water, going back to water. I think we've covered this slide last time. I believe I talked about hydrophilic and hydrophobic already, and the polarity of water and dissolving salt, for example, in water. Water being a good solvent. Any questions on any of this? Okay. Solutions. As you will realize when we start talking more about the concentrations of different solutions and diffusion and osmosis a little bit later in the next chapter. You know, every solution basically is a mixture of something and water. If we were to look at a starch solution or a sucrose solution, salt solution, it's that substance mixed with water. Saline IV. You've heard of saline IV? A saline IV is salt and water. Right? Not to the concentration of salt water in the ocean, but still a mixture of salt and water. And typically in a solution, when we mix those substances together, salt and the water, the sugar and the water, the one substance, crystals, for example, will dissolve nicely within the water so that after a period of time, you won't see those crystals floating around anymore. If you take your coffee and you add sugar to it, when you stir it up, after a few minutes or so, you really can't see the sugar crystals anymore. They've dissolved within that coffee, within the water of the coffee. So the particles are evenly dispersed, and typically, if I didn't have coffee in there and I just had water and sugar, that solution would be clear. I will show you that the solute, the sugar or the salt, whatever it is, has dissolved. Solvent is water. It's what's doing the dissolving. For our sake, yes. If we're going to talk about stripping the floor, some chemical, then it would be different, but yes. So we've got the solvent, which is water. We have our solute that's being dissolved by the water. If we mix the two together and you no longer see the solute, can't tell it was there, then it's a solution. Colloid. A colloid is, it's a mixture of different things. There could be water there. There could be, will be other substances as well. But you can see that there's other substances there. If you shake that substance or you try to hold it up to the light, you can see particles floating around in it. You can't see right through it like you would be able to with a solution. We say that colloid has the ability to scatter light. You're not going to be able to hold up a glass of milk, for example, and look through the milk. The more water content in the milk, like skim milk, it might be a little easier, but whole milk, you're not going to be able to hold it up and look right through it. So that's considered a colloid. The particles don't totally evenly disperse. You can still see that there's particles floating around. And then lastly, a suspension. Think of cells or substances being suspended in a more watery solution, more watery environment. Best example of this is your blood. Ever gone to get blood sample done? When they take the blood out from your vein, put it in a test tube, it looks nice and kind of dark red, right? But if you were to let that tube sit for a few minutes, it would separate into layers. Not only would it get thicker, but it's going to separate into layers. And you will see a layer of red blood cells, you'll see a layer of white blood cells and platelets, and then you'll see a nice kind of clearish layer at the top of that, which is the plasma. Plasma of your blood is mostly water. So what's in the plasma that's suspended? All of those cells, the red blood cells, the white blood cells, platelets. They're all suspended. You cannot see through it, right? You're not going to be able to see through it. But think of it as the most dense of all of three. We have a certain amount of red blood cells, a different amount of white blood cells, different amount of platelets, but they're all within this watery plasma environment. It's a suspension. Does this make sense? Yes. All right, pH scale. Notice the way I wrote pH. I did not make a mistake. The P is always written lowercase, and the H is always written as a capital H. Right? It's the power of hydrogen. We are looking at the hydrogen ion, concentration of the hydrogen ion within a particular solution. How much hydrogen ions are there? When I say hydrogen ion, I'm talking about the hydrogen atom that has a positive charge. Why would the hydrogen atom have a positive charge? Good, because it has lost an electron. Okay. Remember we were saying before it loses an electron in hydrogen? That's what we were talking about. Well, this hydrogen ion is essentially just a proton by itself. The nucleus of hydrogen, and in that nucleus there was one proton, and how many neutrons? 0, right? And then we have the electron orbital with the one electron. But we don't have that. We lost it. Gave it up. Some oxidation reaction. We don't have it. We are looking at just the concentration of the hydrogen ions in a solution. That's what's going to tell us the pH. You might think of it as, is it acidic or is it basic? If you've ever tasted vinegar by itself or lemon juice by itself, any citrus fruit has an acid taste to it. It's an acid. But something more basic, like milk of magnesia, cream, sour cream, things like that, have a different taste to them. Okay, a more alkaline, a more basic taste to them. So we can describe an acid as having a high concentration of these hydrogen ions. If we have a high concentration of these hydrogen ions, food, that solution, that substance is acidic. It's an acid. If we have a low concentration of these, then the opposite is true. The substance is a base. It's a base. So we have a scale. On one end are the acids with the high concentration of hydrogen ions, and on the other end are the bases with the low concentration of hydrogen ions. What's in the middle? Okay, neutral. Something that's neutral, like pure water. The chemical formula for water is H2O. Okay, if I take a hydrogen ion out of that, then I'm going to end up with an oxygen and a hydrogen. Right? I've got an OH over here and an H over here. Similar to what I was saying before about the dehydration synthesis reactions, right? Where I have to take an H from one A and an OH from the other B in order to put the two substances together. Okay? So if I split water like this, I have hydrogen ions on one side, that's my acid, and I have the OH, this is called the hydroxyl functional group, or often hydroxide, that's the base. So here's my scale. I have acid, high concentration of hydrogen ions on this side, and base, high concentration of hydroxide ions on this side. See what I mean? So if I have a high concentration of hydroxide ions here, then I also have a low concentration of hydrogen ions there. If I have a high concentration of hydrogen ions on this side, then I have a low concentration of hydroxide ions. And in the middle, when you're ready, but I have neutral substances. So the neutral substance is water, pure water. How would you describe the concentration of hydrogen and hydroxide ions in water? Neutral. They're equal to each other. They're equal. Concentration of hydrogen ions is equal to the concentration of hydroxide ions. The brackets just mean concentration. one hydrogen ion, I have one hydroxide, I put them together, I get water, which is my neutral substance. pH scale will go from 0 to 14. The closer you get to 0, the more acidic the solution is. The closer you get to 14, the more basic the solution is. The ends of the scale, the stronger the solution. Either the stronger the acid or the stronger the base. Closer to the middle, closer to neutral, the weaker the solution is. So if I have a solution that is pH equal to 6.4, what would you say it is?
More acidic.
Okay, it's an acid.
A weak acid.
And since neutral in the middle, I told you 0 and 14 were the ends, right? The middle is 7, obviously. So if you look at 6.42, that is definitely an acid. It's lower than 7, but it's closer to that neutral side, so it's considered a weak acid. What if I said pH equal to 13.9? It's a strong base. It's a strong base. And that means that there's a lot of hydroxide ions there and very little hydrogen ions. Blood. It has a pH, I gave it to you over here, it's a range, but approximately pH 7.42. 7.42. Blood is a weak base. People think, oh no, blood's neutral. It's got to be neutral. Nope. It's not. Right? There is a range. It's 7.35 to 7.45. Either way you look at it, it's still a base. Still a weak base. If the pH of your blood is below 7.35, meaning if it's 7.34 or 7.33 or 7.3, then the blood is too acidic. And that's a condition called acidosis. You're going to be in the hospital, in other words. In that.
Things like skin to the vagina, like the fluid is
Mhm.
more acidic.
Great. Yeah. pH of different fluids in the body is either meant to help you with digestion or to help your immune system to kill pathogens that might enter. It's a protected mechanism. So the pH of a woman's urinary tract, for example, or reproductive tract is more acidic to prevent her from getting an infection. Contrary to that, we were talking about the reproductive system, a male semen is more basic. So it's counteracting the acid environment that it's going to go to. It's protecting itself. The semen around the sperm being more basic are protecting the sperm from the acidic conditions they're going into.
So what are probiotics?
Probiotics are generally bacteria that act like antibiotics that help your body to fight off infections, build up enough of the good bacteria within your digestive tract to help you digest food properly. No, not necessarily. No, just basically organisms, bacteria that are good for your digestive health. You have bacteria within your intestines and within your stomach that can help you digest and also prevent you from feeling comfort due to ulcers, for example. So probiotics are one of those kind of good groups.
So are they like enzymes?
Are they like enzymes?
Yeah.
They can produce enzymes, certainly, that will help you digest things. It's like eating yogurt. There's bacteria in yogurt. You have bacteria on your body and in your body all the time, right? Bacteria are not bad, generally. They're not. They're helpful to you. You wouldn't be here without them. Okay, honestly, you wouldn't. You wouldn't be able to properly digest what you're eating without bacteria within your body. So you have bacteria on your body, in your body all the time. certain bacteria, like the probiotics or like the bacteria found in yogurt, for example, help to make the balance between the good bacteria and the bad bacteria, or the yeast that's also in your body, to kind of keep those organisms in check. So if you take antibiotics for a long period of time and you kill the bad bacteria that might have given you the initial infection, but also those antibiotics kill the good bacteria, that's normal flora to your body, you can end up with another infection as a result of that. Has that ever happened to you, anyone? Right, or someone you know? They've taken antibiotics for a bacterial infection, then they get a fungal infection, right? They get thrush, yeast infection in their mouth, or they get a yeast infection in the vaginal tract. Yeah, sure. Eyes, there's a lot of different things that can, yeah, a lot of different things that can happen. All right. So, but what we want is, we want the normal bacteria to be there at a good enough level to keep the other organisms, the bad bacteria, the yeast, and so on, to not get so high that it causes us discomfort and some other situations we don't want to begin with. Questions on the pH scale?
So if a molecule is going to have something H2, H3, usually it will be acidic?
Probably. Yeah. More likely you'll see the H at the beginning of the chemical formula, and that's a single acid acid. If you see an OH at the end of the chemical formula, it's a signal it's a base. Oftentimes you can tell just from looking at HCl. Does anyone know what HCl? Okay, yes, you already know it. But HCl is hydrochloric acid. The H in the beginning is a signal, a sign to you that there is a lot of hydrogen ions there. And the acid in particular, because it has a lot of those hydrogen ions, is considered a proton donor. Hydrogen ions are essentially protons, and these acids have a lot of them, and they're willing to donate them to other substances. Whereas a base is considered a proton acceptor. So if I have sodium hydroxide, the OH at the end of that, it's a sign to you that that's a base. And this sodium hydroxide can accept a proton, maybe from hydrochloric acid or something, and we would end up making water and sodium chloride. So, it's kind of like they swap dance partners.
So for the redox reactions, are the reductions usually acids because it has the H in it?
Yes, but we won't describe them like that. I think I might have some examples on the next slide. Do I? Yeah, I do. All right, well, we've done a few examples already. So you should be able to answer these on your own. If you have trouble, then ask me in recitation, but I think you'll be okay. Or next class. Everyone get the idea? Yes. So buffers. How many of you have taken chemistry? Maybe in high school, or so you might have worked with a buffer. And I'm not talking about the buffer like the going to buffer your car, buff your nails. A buffer resists change in pH. So let's say I have a solution and the pH of my solution is 2. In order for the experiment that I'm doing to work, I need the pH of that solution to stay at 2. But in order for my experiment to work, I have to add sodium hydroxide to it. If I add sodium hydroxide to my solution that has a pH of 2, what's going to happen to the pH?
It's going to go up.
It will change, and it will increase, right? It will move from 2 maybe to 3, maybe to 4, maybe even up to 8, depending on how much sodium hydroxide I'm adding. Then my experiment's, in the garbage. It's not going to work. So I could add a buffer. a particular substance that will combine with these hydroxide ions, these extra hydroxide ions, and get them out of the way. So that the pH will stay at 2. And I would still be adding my sodium hydroxide in place. So the buffer resists a change in the pH. And in terms of chemistry, we would have a buffer for all different pHs. We could work with a pH 1 buffer, or a pH 7 buffer, or a pH 9 buffer, depending on what reaction we were performing and what we wanted our pH to stay stabilized at. In the body, you have buffers as well. There's phosphate buffers, carbonic anhydrase buffer system, carbonic acid buffer system that you can see here. So you have these buffers already within your body. If the pH of your blood, that I said before was about 7.42, goes a little bit too acidic, 7.3 or so, or a little bit too basic, then the buffer systems within your body act just like those in chemistry to combine with the extra hydrogen ions or to combine with the extra hydroxide ions and put the pH back where it's supposed to be. By the way, your pH between 7.35 and 7.45, that range, what's that called again? It's that term that I said I would mention at every class. That starts with an H. Homeostasis. Right, so one example of homeostasis within your body is keeping the pH of your blood within that range. That's your tolerable limit. If you go below that or above, things are going to happen. It could be lethal. It could be deadly to you. So we have buffer systems to help to protect us if the pH of our blood changes. We'll talk more about the buffer systems in a later chapter as well. Organic compounds. This is the next major section for chapter 2, dealing with the organic compounds. Did I lose my eraser? Oh, here it is. I think we talked a little bit about the term organic already, didn't we? When a substance is organic, what atom does it contain? Carbon. Good. Carbon, we said carbon is the element of life, one of the building blocks of life. Carbon bonded to a hydrogen covalently is our basic building block. Carbon covalently bonded to hydrogen. That's what we are going to look for when we say a substance is organic. And this organic is totally different from the organic vegetables that you go to the, Whole Foods to buy. That organic definition is different from this one. That organic definition is maybe they've grown those apples, that produce, whatever, without using pesticides for 3 years. That might be their definition of organic. Ours is it contains carbon covalently bonded to hydrogen. Okay. If we do not have carbon, or if we don't have carbon covalently bonded to hydrogen, then the substance is inorganic. It's not organic. We were looking at water, H2O. There's no carbon there. So H2O, water, is inorganic. It's not an organic substance. Give me another example, because we talked about a few of them already today. What's inorganic? Okay, I heard CO2, that's good. CO2 is considered inorganic. There's carbon, but there's no covalent bond to hydrogen. Good, that's a tricky one sometimes.
Hydroxide.
Okay, hydroxide. What else? Salts. NaCl. I could even put sodium hydroxide here instead of just hydroxide. But all of those substances are inorganic. we're looking at what can we take carbon and hydrogen and, you know, put together in all different ways to make within our body. There's 4 major groups of these organic molecules. Carbohydrates, lipids, good, proteins, and nucleic acids are the 4 groups. In each of those, there's carbon covalently bonded to hydrogen. They're all organic. We've already looked at carbon and the arrangement of the electrons and the protons and atomic number and mass number and all that. So are there questions on any of this? You should remember from last class, carbon can bond to up to 4 other atoms. We can take 4 hydrogens and covalently bond them to 1 carbon. You can take other atoms too and covalently bond them here.
Is that because they have 4 electrons?
Yes.
So if it was 5, it would be they combined.
It would be different, yes. Everything in our lives would be different, basically, yeah. So there's 4 spots here, there's 4 empty seats in this outer electron orbital that we can fill. And the most that we can have bonded to this 1 carbon is 4 other atoms. We can have less than that and still be stable. But we really can't. We cannot have more. All right, so here are a chain of carbon atoms bonded to each other. This red circle is a carbon atom, and it's bonded to a hydrogen on this side, hydrogen over here, hydrogen up here, and at the bottom, it's bonded to another carbon. Essentially, there's 4 lines coming away from that 1 carbon atom. If I look at every one of these carbons, there should be 4 lines coming away from it. Does this make sense? So this molecule, since it is composed of carbon and hydrogen only, we call a hydrocarbon. It's a hydrocarbon chain. Hydrocarbon chain. Hydro for hydrogen, and obviously carbon. By the way, I would not have you draw anything like this. But if I show you this, you should recognize that it's a hydrocarbon, that these lines are covalent bonds. Single covalent bond. This is another diagram. Okay, each of the C's is a carbon atom. Here they're not showing you, but we are going to assume that there are hydrogens at the end of all of these branches. And if we count up the number of lines extending from each carbon, there should be 4. 1, 2, 3, 4 for that carbon. And this shows you that carbon can not only branch, can have a chain and branch off the chain, but it can also form a ring. It can coil back and bond to itself, form a ring form. This is the primary form that we'll be looking at when we look at a monosaccharide, a simple sugar, like glucose or sucrose. We will be looking at rings. Ribose or deoxyribose, we'll be looking at a ring form of carbon like this. And this is an even more simplified diagram of a carbon ring. They're not showing you any C's or H's or anything. You just have to assume that they're there. The lines, there's 1 C, 1 carbon here, and another carbon here. They're bonded together. You're assuming that there's hydrogens off of that. They don't have to show it to you. Carbon here and a carbon here, bonded together, and so on. Really, from now on, you'll probably be seeing diagrams more like this one than you will like this or this. If there was anything here other than carbon, they would show you that. For example, you might have an oxygen. You might have an oxygen over here, and it would show you the oxygen like this. It would actually have the oxygen written within that ring. That means there's not a carbon there. Instead of a carbon, there's an oxygen. or there might be an OH group off of this carbon instead of just the hydrogen that we're assuming is there. It would show you that. Oh, functional groups. I already kind of talked about them a little bit, but in your book, I believe page 42 in your book has a table of the functional groups listed on it. Upside down, right side up. Table 2.5, page 42. There's many functional groups listed here. Bless you. Okay. You can see from each of the functional groups that there are essentially combinations of different atoms. It could be an oxygen and a hydrogen, or it could be 2 oxygens and a hydrogen, or it could be a nitrogen and 2 hydrogens. Just combinations of atoms that we typically see attached to a carbon atom, either in a hydrocarbon chain or in a carbon ring structure. So groups that often go together, they're functional groups. The ones that you need to know, you don't need to know every single one on there. Robbie, I know that's going to be one of your questions, right? Okay. You don't need to know everyone on there. However, you do need to know the hydroxyl functional group, which is the OH. See the R there? There's no element that's R that we'll be talking about. The R means that there's just something there for the OH to bond to. It could be just a carbon atom, or it could be a chain, or it could be a ring, but that OH is bonded to that R, that different substance. R is like anything. So OH is hydroxyl. Let's see. Carboxyl, you need to know. Carboxyl is the COOH. Often we will call this the COO group. COOH. Or you might see COO minus. Hydrogen, I know, took its proton and it left. Both of these are called carboxyl. It's the carbon double bonded to the oxygen, bonded to another oxygen, and maybe there's a hydrogen here and there might not be. This COO group. And this group is bonded to something else, some R. So that we have 4 lines extending, right, from the carbon atom. Carboxyl groups, you will see when we talk about proteins. You'll see carboxyl groups in proteins. The hydroxyl I had mentioned earlier, the OH group, you'll primarily see in carbohydrates when we look at sugars, also alcohols. Okay, towards the bottom, second one from the bottom, can't really see it on the screen, but down here at the bottom, the last 2, phosphate group and the amino group. You need to know those 2. Phosphate group you will see when we talk about ATP, which is energy to us. And you'll also see the phosphate group in nucleic acids, DNA and RNA. The amino group, which is the last one, it's an NH2 group or an NH3 group. The amino group you will see in proteins. Amino and amino acids make up proteins. You'll see them in proteins. So be able to spot these functional groups. As we go through the 4 types of organic molecules, I will point some of them out to you, but you should as well be able to spot them and name them. Question, no? Yes, no.
These two last,
Sure, the last ones, yep.
they're not carbon.
They are. They are all bound to carbon. Right, so the R that you see here on all of them, the R is a carbon atom from something. But some of the groups have another carbon within the group. Like the Cu group or the ester or the carbonyl, they have another carbon atom within the group itself.
So R is always carbon?
R is carbon. Yeah. It's carbon with something else to it.
R is also, so in hydroxyl, the R will also have carbon?
Carbon. The R is carbon. I'll show you what I mean.
Why doesn't it have?
See, I know. Because the R can be so many different combinations of things. It's just easier and more simple to write R. If you remember the structure of the amino acid, remember this from Biology 5, where the amino acid had a central carbon atom, an R group, a hydrogen, a carboxyl, an amino. The R could be anything. It's kind of like that. So it's just the letter that we use when something is varied. It's a variable. I'll show you what I mean. Okay, so at the very top right here, there's a chain of carbon atoms. And look what's coming off of the chain. OH, hydroxyl functional group. Yes? Same thing down here, hydroxyl functional group. But each of them is bonded to a carbon. But the carbon is not part of the hydroxyl functional group. Hydroxyl, hydro, and oxyl. It's hydrogen and oxygen only. The name of the group will help you remember what's in it. Carboxyl, the carboxyl group that I was drawing here. What's in the carboxyl group? Carb, carbon, and oxyl, oxygen. I'll show you another one here. Bear with me because I'm flipping, so just so at least you can see what I mean. This is a steroid at the very top, just like testosterone or estrogen. If you look at the top, you see the carbon rings? Just like that drawing earlier in orange, you're assuming there's a carbon here, and a carbon here, and so on. This carbon has a hydroxyl functional group off of it. Groups of atoms that we typically see. Here's a protein. Be easier to see on this one, I think. COO. What group is that? It's the carboxyl functional group, and it's attached to another carbon. And then same thing over here. This carbon has an amino group attached to it. So you should be able to spot these groups as we go through. You will not have to draw any of them, but if I ask you what this group is, you'd be able to name it, recognize it. So looking at the 4 different biological molecules, let's talk first about the carbohydrates. These are sugars, saccharides. The simplest of the carbohydrates are the monosaccharides. One sugar, one simple sugar. Monosaccharides like glucose, like ribose, deoxyribose, galactose. All of these are monosaccharides. One simple sugar, meaning one chain or one ring of carbon, with hydroxyl functional groups coming off of it. The hydroxyl functional groups, I showed you that picture earlier, the hydroxyl functional groups are what make the carbohydrate polar. They're what make this carbohydrate able to dissolve in water. The more OH groups, the more easily that carbohydrate's gonna dissolve in water. Remember, like dissolves in like. Water has oh, I erased it now, but
This is
water has
hydroxyl.
hydrogen and OH.
Hydroxyl.
OH, like dissolves in like. If you were to count up the number of carbons, hydrogens, and oxygens in a monosaccharide, this is glucose here, you would see that there's a 1 to 2 to 1 ratio. That means for every 1 carbon in a monosaccharide, there's 2 times that number of hydrogens, and there's the same number of oxygens as carbons. So it's 2, yeah. So it's a 1 to 2 to 1 ratio. The formula for glucose is C6H12O6. That holds true, right? For every 1 carbon, there's twice that number of hydrogen and then the same number of oxygens to carbon. 1 to 2 to 1 ratio. Notice that you can see the oxygen right here in that carbon ring. That means there's no carbon here, there's an oxygen instead. But there is a carbon here at this point, at this point, at this point, and so on. And the little numbers, if you can see that in your copy or in the book, they're very small. The little numbers represent the carbon atom at that point. They number the carbon atoms. So this is the first carbon. It has a hydroxyl group off of it and a hydrogen off of it. And it's bonded to another oxygen on this side and a carbon on this side. That's the first carbon right there. There's the second carbon, and the third carbon, and the fourth carbon, and the fifth, and the sixth isn't even a part of the ring. It's outside the ring. You all with me? Sixth carbon is this one up here. The carbon CH2OH. That's the sixth carbon in the C6. So you can see where all the 6 of them are. And we could count the number of hydrogens there. We should get 12. We could count the number of oxygens. We should get 6. You will not have to draw this, nor will I ask you, is this glucose or fructose or galactose or anything like that. But you should recognize it's a carbohydrate. And there's a carbon ring, and there's hydroxyl groups, and it's definitely going to dissolve in water because of those. It's a monosaccharide, things like that.
So do all monosaccharides have that 1 to 2 to 1 ratio?
The word all is a difficult one, right? But the ones that you'll be looking at, yes, they will. Yeah. It's true of other saccharides as well, but most true of monosaccharides. All right, so you can see the examples of a few of the monosaccharides I just mentioned and compare them here. As I said, I would not ask you to identify them as this is galactose, whereas this is fructose, and that's glucose. I wouldn't ask that. But realizing that they're all monosaccharides, you should know. The galactose, you can see the arrangement of the carbon atoms is different than fructose and also glucose. If you were to count up the number of carbons, 1, 2, 3, 4, 5, 6, and hydrogens and oxygens in each of these, it's the same. You take 2 eggs and you add a cup of flour and a cup of sugar, and it's the same ingredients, but you put it together in such a way that you come out with different products. These are called isomers. Previous slides. Isomers. They have the same chemical formula. All of them. C6H12O6. But the arrangement of the atoms is different for each one. They're isomers. We are going to talk more about deoxyribose and ribose when we get to nucleic acids. But deoxyribose and ribose are monosaccharides. themselves, they are simple sugars. Look at the endings of all of these examples I'm showing you. At the end, deoxyribose, ribose, glucose, right? They all have an OSE ending. OSE at the end. That's a signal that that's a saccharide, that it's a sugar, that it's a carbohydrate. So even if you don't know lactose or maltose, just from the name and the OSE ending, you know it's a carbohydrate, it's a sugar. So look for that ending. How is glucose different from sucrose? So here's glucose over here. It was a monosaccharide. How is it different from sucrose? Right, good. Sucrose is a disaccharide, or sucrose is two monosaccharides that have bonded to each other. In order for us to make sucrose, to bond the A and the B to get our C, what do we have to do to A and B? Good. We got to dehydrate them. We got to take water out. Dehydration, synthesis, anabolic reaction. We're taking the OH from here and the H from here. We're removing this water, that's our byproduct, in order to put these two monosaccharides together. Now this carbon is lonely, and this oxygen needs something else to bond to. So the carbon over here is going to bond to this oxygen. And you'll see that link right here. We have now synthesized sucrose, our disaccharide. Two rings, two monosaccharides bonded together. If I wanted to digest sucrose, what do I have to do?
Add water.
Add the water back in. So I'm going to just go this way. I'm going to add the water to sucrose, and then I get back my glucose and fructose again. And that would be hydrolysis. I'm adding water back in to break the bond. Break the bonds within water and break the bond within the sucrose molecule.
So is lactose, glucose like lactose, but without the water?
Yeah, no, lactose is essentially just like sucrose, and your body is able to digest it because of the enzyme lactase. If you don't have the enzyme lactase, but you would need water also, if you don't have that enzyme, you wouldn't be able to break this bond. So it's not only requiring water, but also an enzyme to help that reaction occur. So individuals that are lactose intolerant, you know, if they eat lactose products, dairy, ice cream, things like that, milk, they would have a very difficult time digesting this, even though they drink a lot of water. It's still going to hurt a lot if you eat a product like that, because they don't have the enzyme lactase. So you might take a pill like Lactate or something that is the enzyme that will help you to digest this substance.
So are oligosaccharides I know they're disaccharides, but could they be more than?
Yeah, they can. So we were just looking at sucrose, which is a disaccharide. It is under the heading of oligo, which means a few. It's a few saccharides. So 2 is probably the more common that we'll talk about, but there could also be trisaccharides that are oligosaccharides. I would say for oligo, 2 or 3. You know, I wouldn't show you really anything other than disaccharides for this. Yeah. But this is considered an oligosaccharide. Mono is just 1. Oligo is a few, maybe 2 or 3, of which the disaccharides are the best example, or just 2.
So if fructose is the main sugar in fruits,
In fruits, okay, yeah.
does that mean that fruits don't have monosaccharide?
No, fructose is a monosaccharide, and that's why fruit tastes so sweet, because they're monosaccharides. The more monosaccharides, the more sweet it tastes.
Okay, so basically anything that's oligosaccharides are basically artificially.
Not necessarily artificial, but there's more of a substance there. saccharides bonded together, so it's not going to taste as sweet to you. As we get to the next category, which is polysaccharides, an example of that is starch. Starch is many glucose molecules bonded together. Have you ever bit into a raw potato? Probably not, but I think you can imagine what it's going to taste like. Not going to be very sweet. All right. The longer this chain of saccharides is, the less sweet it's going to taste. The more you're going to have to digest it to taste the sweetness. If you just take a spoonful of sugar, it's pretty sweet. If you chew a cracker that has some sugar in it, but is not a sugary cracker, Ritz cracker or saltine, the longer you chew it, the more sweet it will taste to you. It's the enzyme within your saliva, salivary amylase, that's helping to break those oligosaccharides apart into monosaccharides. That's happening in your mouth, just when you're chewing. It's one of the first processes of digestion that's occurring. And as you're breaking those groups of saccharides into smaller groups, you're starting to taste more of the sweetness to it. Coming from someone that's eaten a lot of saltines. If you have never done that, take a saltine and chew it within your mouth for a while, longer than you would normally chew something, and see if the sweetness changes to you. At first it's going to be salty, but then it should turn sweet and more sweeter as you chew it.
That's what's.
Same thing. Same thing. Okay, questions on the oligosaccharides or monosaccharides? You can see lactose here, we were talking about that before. This is again the bond that's difficult to break in someone that's lactose intolerant. Maltose is another example of a disaccharide. And then the polysaccharide, like the starch I was just mentioning to you, is a chain of many glucose molecules, 1,000 glucose molecules bonded together. Each of these purple hexagons, I should say octagons, each of these purple shapes is one glucose molecule. It's one monosaccharide. So we've got one bonded to another, and it keeps going. So we have maybe 1,000 of them, and that's a starch molecule. And you have a lot of those in a white potato. So carbohydrates are a good energy source. Why? Because what am I going to do with this carbohydrate? I'm going to break it down to get monosaccharide like glucose, and then I can use glucose in cellular respiration to break it down for ATP. So they're a good energy source. Yeah.
So the reason why complex carbohydrates are better than simple carbohydrates
Yep.
is because it breaks it down slower than it would a simple carbohydrate?
Correct. And it's going to last longer, and you're going to feel full longer, rather than that sugar rush that they say. I don't know if it's ever really been scientifically proven, but yeah. You know, if you eat a bag of Skittles, it's not going to fill you up very long. Whereas if you eat a bowl of pasta, like that. Whole wheats, whole grains, obviously is better for you than white rice, white potatoes.
So are the whole grain things polysaccharides?
Still polysaccharides, but it hasn't really been chemically processed and bleached to the point where it's white. Anything that you have that's white is really not naturally white. It's been bleached, passed through animal bones, things like that, to get to be white. The coating around it, like the seed coatings and things that would be healthy to you, have been removed to just make it look more pretty. Sorry. Sorry. Okay, so any questions on the carbohydrates? Yes.
So polysaccharide only glucose, no?
Polysaccharides?
Only glucose?
Chains of glucose. Yeah. And in the case of, if we were looking at a plant, then we would be able to store that glucose as starch within a plant, like the potato. In our body, we're not storing starch. Okay? We will store glucose as glycogen. And glycogen is another example of the polysaccharide. Glycogen is stored, for example, near our muscles. Where our muscles need a constant supply of energy, we have these little glycogen granules. these little groups of glycogen, which are glucose molecules all bonded together, and we break them apart when we need them.
Lactate,
Lactate.
in muscles?
Lactic acid can be produced if you overuse your muscle, and instead of inputting enough oxygen to perform aerobic respiration, then you kind of switch to an anaerobic method of which you're going to produce lactic acid. So, there's a certain amount there, but it's not helping you, in other words. The presence of the lactic acid around your muscle is going to make your muscles sore. It's going to hurt. You work out to the point where you're sore the next day, then you know you've got lactic acid around your muscles. For that short period of time that you were working out, it was like the muscle's last resort to get energy, was to make this lactic acid. So polysaccharides, oligosaccharides, monosaccharides are all carbohydrates. You can look for the OSE ending, but glycogen and starch don't have that. So those you're just going to have to remember. Those are polysaccharides, those are carbohydrates. Okay. We will stop here and then pick up next week and finish up the macromolecules. We'll do proteins and lipids and nucleic acids. That should be the rest of chapter